Hello everybody, I'm Samin Abulhasanzadeh. I'm as a scientist in Merrick's Nutrition Science in Ireland. Thank you very much. Give me this opportunity to share my experience. The topic of this presentation is production of lung cancer antibody as a nanopharmaceutical products. This presentation has three steps: introduction, methods, main result, and conclusion. I'm gonna explain introduction step. This present invention is related to the fields of medical biotechnology, nanobiotechnology, and nanomedicines. Is especially designed for the targeted treatment of lung cancer. In this context, nanomedicine have been considered as advanced tools for medical and chemotherapy treatments. Due to their unique properties, including nanoscale size, wide range of biological activity, and ability to precisely target, nanomedicine have been widely used in the treatment of various diseases, including cancer. In this invention, the nanomedicine product with the ability of controlled release of HOCl at the tumor site is presented as an advanced and new technique for the treatment of lung cancer. As a strong antimicrobial anti-tumor agent, HOCl can effectively affect tumor tissues. While it affects healthy tissues less, the use of nanomedicines for the controlled release of HOCl helps to enable targeted therapy and reduce the side effects of non-targeted therapies. Nanodrug formulation that is able to carry and release HOCl at the precise location of the tumor. Precise control over the amount and time of HOCl release to optimize the therapeutic effect and reduce damage to healthy tissues. Using special nanomaterials to improve drug properties and tumor diagnosis, considering that lung cancer is one of the most common and deadly types of cancer. The nanodrug product with controlled release of HOCl can be an efficient and advanced treatment tool to improve the treatment result and quality of life of lung cancer patients. Step 2: methods. HOCl nanodrug product with controlled release capability in the tumor site of lung cancer treatment is especially used in medical and pharmaceutical industries. As one of the most common types of cancer, lung cancer accounts for approximately 40% of new cancer cases worldwide. Considering the importance of HOCl in cancer treatment and potential benefits of its delivery by inhalation, the development of new drug delivery systems that can deliver HOCl in sustained and controlled released manner and minimize the side effects resulting from it is very important. This nanoparticle has a two-layer structure. Inner layer including alginate with perfluoropentan, PFP, which acts as a phase change agent, and calcium hypochlorite, calcium OCl2, as a source of hypochlorite anion HOCl- is located in the nanoparticle alginate core outer layer, including chitosan coating conjugated with fluid and salicylic acid. The fluid present in the chitosan coating helps the effective targeting of the nanoparticle to the cancer cells, as the salicylic acid acts as a proton source. The production process of this nanoparticle uses a two-step method including nanoemulsion and coating using ionic gelation technique. After immolate nanoparticle delivery to lung, the nanoparticles are specifically targeted to cancer cells using the properties of fluid on the surface of chitosan. After targeting, the radiation of ultrasound waves changes the phase of perfluoropentan and increase the internal pressure of the nanoparticle, which leads to the collapse of the nanoparticle structure and the release of HOCl. This HOCl is released with a certain concentration to the tumor site and induces apoptosis and eventually destroys the cancer cells. By using this advanced drug delivery system, it is possible to achieve more effective lung cancer treatment by reducing side effects and improving the efficiency of HOCl delivery to the tumor. We wanted to talk about charts. Result step. Figure 1 shows the release profile of HOCl from HOCl PFP alginate chitosan nanoparticles using ultrasound waves in 350 minutes. Figure 2 cell viability diagram of A594 cells treated with HOCl PFP alginated chitosan NPS for 24, 48, and 72 hours. Cell viability diagram of A549 cells treated with HOCl for 24, 48, and 72 hours. Our data are presented as mean plus minus SD and sign 3. Figure 3. Western blot analysis of caspase. Caspase 3 PARP minus 1 BAX BCI2 at 24 and 48 hours after treatment of A594 cells with HOCl PFP alginate chitosan NPS. Data are shown as mean plus minus SD and 6.3, P less than 0.05, P less than 0.01, P less than 0.001. Figure 4. Relative tumor volume analysis in tumor-bearing mice that were treated. And second 3. Survival analysis of mice under different treatments. And second 5. Weight of tumors isolated from tumor-bearing mice on day 10. And second 5. Eventually, we talk about the conclusion step. The invention of HOCl PFP alginate chitosan NPS has significant industrial and medical applications. particle with a double layer structure that includes alginate containing PFP and chitosan conjugated with fluid, and made using advanced production techniques such as nano emulsion and inoculation, is specifically designed for the treatment of lung cancer. The industrial advance and application of this invention are target treatment of lung cancer, targeted drug delivery through inhalation, increasing the effectiveness of treatments using ultrasound waves, application in medical centers and hospitals, application in cancer research centers. Explaining the advantages of the invention compared to previous inventions. Number 1. Targeted pulmonary delivery of HOCl. Number 2. Sustained production of HOCl at the site of cancer. Number 3. The possibility of a nano-drug inhalation administration. Number 4. Significant reduction of drug side effects. Number 5. Being fast-acting and destroying lung cancer cells. Number 6. Accurate spatio-temporal control of drug release. Number 7. Control of drug delivery cycle. Number 8. Increasing therapeutic effectiveness through pulmonary drug delivery. Releasing the drug and increasing the efficiency of penetration into the tumor through the mechanical effect caused by radiation of ultrasound waves. The invention of HOCl PFP alginate chitosan NPS has significant industrial and medical applications due to special abilities in the targeted treatment of lung cancer, improving drug delivery methods, and increasing therapeutic effectiveness. This nanoparticle plays an important role not only in clinical and hospital treatments, but also in scientific research and introduced as an advanced tool in the fight against lung cancer.